Welcome to the Fatty Acid Forum, sponsored by Virtus Nutrition. Let's listen as Dr. Charlie Staples from the University of Florida answers the question, how much essential fatty acids can escape biohydrogenation in the rumen?
In the last webinar, we indicated that essential fatty acids, namely linoleic and linolenic acids, are present in most feedstuffs for dairy animals, although the concentrations from feedstuff to feedstuff can vary widely. Linoleic acid, the omega-6 fatty acid, is usually present in much greater concentrations than linolenic acid, the omega-3 fatty acid. And although these fatty acids are present in nearly all diets, the amount of these essential fatty acids that escape intact to the small intestine for absorption is relatively small. The purpose of this webinar is to briefly describe how the essential fatty acids are metabolized in the rumen, what factors may increase their delivery to the small intestine, and how much of the dietary essential fatty acids may actually end up in the small intestine. Linoleic acid, C18-2, and linolenic acid, C18-3, exist in most feedstuffs in a tri- or diglyceride form, as described in the previous webinar. That is, there are 2 or 3 fatty acids in one molecule. And once the tri- or diglyceride is consumed by the cow, the ruminal microbes produce enzymes such as these lipases, galactosidases, and phospholipases that cleave the individual essential fatty acids from the glycerol molecule. And this occurs very quickly, usually in a matter of minutes. Now that the fatty acid is free from the glycerol backbone, the ruminal microbes begin a process called hydrogenation, which is simply adding hydrogen molecules to the places in the fatty acid structure where there are double bonds. Because this hydrogenation process takes place in a biological system, it's called biohydrogenation. The steps in the biohydrogenation process involve both isomerase and reductase enzymes produced by the microbes. In the case of linoleic acid, the isomerase enzyme first causes a rearrangement of the naturally occurring structure of double bonds from a cis-cis to a cis-trans configuration. The second step is for the ruminal microbes to change one of the double bonds between the carbon molecules to a single double bond to create this trans 11 C18-1 from the cis-9 trans 11 C18-2. The reductase enzyme again works to remove the final double bond to create stearic acid, C18-0, 18 carbons, no double bonds. And this last step is sensitive to the acidic pHs that we find in the rumen, so that less C18-0 and more C18-1 trans 11 and other trans monoenes such as trans 10 and transdienes such as the trans 10 cis 12 exist in the rumen fluid when the pH drops to approximately 6. So the lipolysis and biohydrogenation of linolenic acid, C18-3 here, involves the same types of enzymes, an isomerase to change the cis-12 double bond to a trans-11 double bond, and then 3 reductase enzymes to eliminate the 3 double bonds of linolenic acid. So you can see that both trans-11, C18-1, and C18-0 are the products formed during the biohydrogenation of both linoleic and linolenic acids. The amount of each long-chain fatty acid consumed in the TMR is not close to the amount of that same long-chain fatty acid that leaves the rumen and arrives in the small intestine. A lactating dairy cow eating a fairly typical diet would consume about 450 grams of 18 carbon length fatty acids in the amount shown here. 20 grams of stearic acid, maybe 110 grams of oleic acid, 280 grams of linoleic, and maybe 40 grams of linolenic acid. The amount leaving would dramatically increase for stearic acid, nearly 19-fold in this example, going from 20 grams of intake to 370 grams leaving the rumen. This is due to the conversion of the unsaturated fatty acids to the fully saturated fatty acid. The amount of the unsaturated fatty acids in the small intestine would all be less compared with the amount consumed. The proportional disappearance of the unsaturated fatty acid in the rumen is greatest for the more unsaturated fatty acid. In this case, only 4 grams is left out of 40 grams fed for linolenic, a 90% extent of biohydrogenation. The cow is only left with 39 grams per day of linoleic acid after consuming 280 grams, an 86% loss. Now we're going to look at some factors that influence biohydrogenation. Those are numerous, but I'll mention just a few in the remainder of this webinar. As indicated in the previous slide, the number of double bonds in the essential fatty acid is important. About 70 to 90% of linoleic acid with 2 double bonds undergoes biohydrogenation, meaning that for every 100 grams of linoleic acid consumed, only 10 to 30 grams of linoleic acid reach the small intestine. Linolenic acid with the 3 double bonds is more extensively biohydrogenated, up to between 85 and 100% biohydrogenation. But why do bacteria work on these unsaturated fatty acids like they do? Some scientists have suggested that this is a protective mechanism because unsaturated fatty acids are toxic to many ruminal microorganisms, and the more unsaturated fatty acid, in this case linolenic with the 3 double bonds, may be more toxic than the less unsaturated fatty acid, linoleic with only 2 double bonds, therefore get more extensive biohydrogenation. Ruminal bacteria incorporate unsaturated fatty acids into their cells, and those unsaturated fatty acids that are incorporated into those bacterial cells may avoid biohydrogenation, and therefore more essential fatty acids travel to the small intestine. So the following study illustrates how fat feeding can influence the lipid content of the bacteria. In this particular study out of France, dairy cows were fed a diet containing no supplemental oil, or one of several diets that contained a number of single different oils. In this figure I'm showing the control diet here and the 8.7% soybean oil diet here in the orange, which is really more soy oil than any cow would normally be fed, but it's a proof of concept study. So the bacteria collected from the solid digesta in the rumen had more than twice the lipid content if the cows were fed soybean oil. Specifically, bacteria were 9% lipid from the control cows and were 21.4% lipid if they came from the cows fed soybean oil. The specific fatty acid makeup of the fat found in the bacteria reflected the fatty acid makeup of the oil fed. The proportion of the 5 major fatty acids are shown in these stack plots for the bacteria coming from cows fed the no soybean oil or a diet containing 8.7% soybean oil. The 2 saturated fats, C16O is blue and C18O is orange. Oleic acid is green, linoleic acid is purple, and linolenic acid is red. Soybean oil is 50% linoleic acid, so you would expect the concentration of linoleic acid to be greater in bacteria coming from cows fed soybean oil, and that is what they reported. Linoleic acid more than doubles from 5.6% in the control cows to 13.5% in the bacteria coming from those being fed this 8.7% soybean oil diet. Because linoleic acid is biohydrogenated in the rumen to a monoene fatty acid, the concentration of C18-1 also doubles, going from about 15.1% in the control animals to 33.2% in the bacteria from cows fed the soybean oil. Ruminal bacteria isolated from dairy cows fed the oils contained oil droplets within their cytoplasm, as can be seen in these transmission electron microscopic pictures. The bacteria on the left is from a dairy cow not fed any oil, and the bacteria on the right is from a dairy cow fed rapeseed oil as an example. It contains large and numerous gray areas. These are probably corresponding to free fatty acid droplets, abbreviated LD as lipid droplets. Ruminal bacteria from cows fed oils all had these oil droplets, regardless of the type of oil. And this may be one way that essential fatty acids can escape biohydrogenation. If bacteria incorporate some of the dietary linoleic and linolenic acids into their cytoplasm and cell wall and then wash out of the rumen, then the fatty acids are delivered to the small intestine. Ruminal fluid pH can have a major influence on biohydrogenation, with increase in acidity reducing extent of biohydrogenation. That is nicely illustrated in this study. in which 5 diets were fed to sheep, in which alfalfa meal, here, was increasingly replaced with dehusked barley. Diet 1 was 96% alfalfa meal and 0% barley. 0% barley. Whereas Diet 5 was 0% alfalfa meal and 64% barley. This changing forage to concentrate ratio is reflected in the decreasing NDF concentration going from 42.8 down to 19.5%, and increasing starch and sugars from 12.2 up to 35.7% of the diet. You'll note that corn oil here made up 2% of the diet across all 5 diets. The ruminal pH values for each diet are plotted along the x-axis in this graph on the left. You can see that the pH was very similar for the first 3 diets, ranging between 6.3 and 6.5. Once the pH dropped to 6.1 in Diet 4, which was the diet of 50% barley, the proportion of the long-chain fatty acids, stearic acid and oleic acid, changed their ranking within the ruminal fluid. Stearic acid is the blue line, and it went from about 50% of the long-chain fatty acids down to about 25% at the lowest pH. And oleic acid went from about 30% up to about 50% at the lowest pH. So corn oil, which is rich in linoleic acid, was likely experiencing less extensive biohydrogenation at the lower pH, and so the completely hydrogenated fatty acid product, stearic acid, was less present in the ruminal fluid. On the right side is a bar graph depicting the biohydrogenation rate of linoleic acid. The hydrogenation rate decreased with increase in acidity in the rumen fluid, going from about 500 milligrams per liter per hour down to about 250 milligrams per liter per hour. This change was likely due to a changing population of bacterial species in the rumen, although they didn't report that. Butyrovibrio fibrosolvens, Ruminococcus albus, are known to be able to hydrogenate polyunsaturated fatty acids and do not grow well at pH values less than 6.0. And other species, such as Strep bovis and Megasphaera elsdinii, often increase in numbers at lower pH values, and they may not have the capacity to hydrogenate long-chain fatty acids. Ionophores may be the most common feed additive used in dairy cow diets across the U.S., and they can have an influence on the biohydrogenation of essential fatty acids. Because ionophores act in an antimicrobial function against gram-positive bacteria, gram-positive bacteria include those that carry out biohydrogenation, so it makes sense that they may affect lipid metabolism in the rumen. In the table on the left, ruminal fluid was collected from a sheep fed a diet of half hay and half concentrate, and then the rumen fluid was added to a substrate of concentrates, some soybean oil, and these 2 ionophores, lasalicid or monensin, in an in vitro study in the laboratory. The fermentation was stopped after 6 hours, and the presence of the ionophores inhibited lipolysis of linoleic acid by 15 to 19%. As a reminder, lipolysis is the freeing of the fatty acid from the glycerol molecule, and it is a necessary first step toward biohydrogenation. Biohydrogenation was reduced also by the ionophores, but by a smaller proportion, this 8.6 to 2.1% reduction compared to the controls. These in vitro results were confirmed by a dairy cow study in which the concentration of linoleic acid in the milk fat increased from 3.4 to 4.3% when monensin was included in the diet, the blue bar representing the cows fed monensin. And in that study, soybean oil was fed at 3.5% of dietary dry matter. The process of heating soybeans prior to feeding also has resulted in less biohydrogenation. At Kansas State University, Reddy et al. Determined that the in vitro extent of biohydrogenation of linoleic acid in soybeans was decreased from 80% in raw soybeans to 58% in roasted soybeans that were roasted at 270 degrees Fahrenheit. In support of this protective effect of roasting soybeans on polyunsaturated fatty acids, it's work carried out by Tice et al. At Ohio State University in which dairy cows were fed soybeans at 19.7% of their dietary dry matter, and these cows had a greater concentration of linoleic acid in the milk when the soybeans were roasted at 289 degrees Fahrenheit, with the linoleic acid being 6.7% of the milk fatty acids compared with 5.5% when fed raw soybeans. And feed intake didn't differ between the two groups. When a calcium molecule is attached to the carboxyl group of the fatty acid molecule in a manufacturing process, creating the dry fat, the extent of biohydrogenation in the rumen is reduced somewhat because the calcium attaches to the carboxyl group of the fatty acid and prevents biohydrogenation. When calcium salts of palm oil were fed to lactating dairy cows at 4% of the dietary dry matter in a University of Illinois study, the extent of hydrogenation of all dietary linoleic acid in the rumen was reduced from 76.7 to 68.7, which was an additional 21 grams of linoleic acid reaching the small intestine. And this effect was similar for linolenic acid as well, but the increase was only about 1.5 grams per day, even though proportions was significantly reduced in the presence of feeding the calcium salt to the fatty acid. To summarize, only 10 to 30% of essential fatty acids in the diet reach the small intestine of lactating dairy cows because of the extensive hydrogenation by ruminal microorganisms. In terms of quantity, this means that only 3 to 15 grams of linolenic acid, maybe 4 to 100 grams of linoleic acid usually reach the small intestine. These amounts can be increased somewhat when the diet contains a greater amount of concentrate, contain an ionophore, when oil seeds like soybeans are roasted prior to feeding, and when calcium salts of essential fatty acids are fed. These are not dramatic increases, but probably significant changes that increase milk fat and tissue concentrations of essential fatty acids.